let's revise a little bit. So in last class we did review, we talked about enzyme, and in enzyme we introduced what is enzyme, and then after that we talked about the properties of enzyme. And I told you, so I'll be talking about this 6 point only in this 4 to 5 class. Okay, all these 6 points. I asked you to remember this 6 point in S3 and R3: speed, selectivity, specificity, reaction condition. Reaction site means reaction site is active site, and regulation. So I told you to remember this cartoon. This is very important cartoon. If you remember, you will understand enzyme better. So these are reaction condition. Just imagine this enzyme is a part of your body, small molecular entity. Functional unit of your body is proteins, and among that protein, enzyme is one of them. So this is a protein, a part of your body. You can even molecular part of body. So you don't tolerate temperature, you don't tolerate that pH. So how do you imagine that your enzyme will tolerate that temperature? So temperature and pressure. So enzyme requires a particular temperature, pressure. It has got teeth. I explained this teeth part is very, very important. What actually this coenzymes part is very important. Coenzyme, in that coenzyme about these vitamins. You have to understand about this vitamin. These are very, very important. This part very, very important. So these 3 slides are very important for formative exam. You know, I will be giving questions from these 3 slides. These 3 slides. So you know which is I will be asking this. I will tell you how question comes. So this, what is the coenzyme? This vitamin B1 or thiamine will form which coenzyme? Niacin will form which coenzyme? You have to remember. Pantothenic acid will form which coenzyme? So you will have to remember that. Vitamin B1 will form which coenzyme? B2 will form which coenzyme? And then which of the following will transfer electron in reaction? Which of the following will transfer electron? I will give 4 among this, and I will put 1 of this. So you will have to pick this. You have to understand which transfer electrons, or electrons means hydrogen, which all group, which all coenzyme transfer electron. You have to understand that. And then biotin, which will transfer CO2, is biotin. Don't forget this. Biotin transfer CO2. You can see here others are there, but just remember for the timing this. And then 1 carbon transfer is done by tetrahydrofolate. 1 carbon transfer is done by tetrahydrofolate. Biotin does CO2. And they are also important. If you remember this 3 slides, you will have very easy memorizing, understanding biochemistry later. So I want you to go revise it many times, repeat it many times, so that you will easily remember. Okay, so these 3 slides are very important. The questions will be coming from these 3 slides. Okay, then we talked about this side chain. What is this side chain? And then after we did all this, so we came to talk about the enzyme classification. And enzyme classification, I told you the enzymes, just consider enzyme as a worker. So they are like worker, and this tiny worker, and they have their specific job, and that is called specificity. Specific reaction is called specificity. We are in classification nomenclature, we are talking about the specificity of enzyme. So each enzyme has got a special specificity. Based on that, they have been given systematic name, okay, based on its function. And common name is also somewhat related to function, but systematic name is related to their class. First they systematically classify the enzyme, and then after classification they put the name. So you gave the name of the enzyme according to its job it performs. So we have classified like that. So this is the enzyme commission number, the number you give to a particular enzyme that actually describes its job prescription. What is its job? So this is enzyme commission number. You can see this. We talked about this. So this is an important point that you need to understand. All oxidoreductases require coenzyme. Most transferase require coenzyme. Ligases, we talked about this. So we have already done oxidoreductase. In the oxidoreductase group of enzyme, you will see the oxidoreductase group of enzyme will come with the one of these 4 names. Okay, so understand, memorize this 4 names. I have given also examples of that. So enzyme is everything. Classification of enzyme is also very, very important. Okay, so more you invest time in enzyme, easier will be later you will have easier classes, so you will understand biochemistry better. So I want you to invest time in there. So you can see somewhere reductase is written, dehydrogenase is written. Okay, so here is written, what is written, dehydrogenase, reductase, dehydrogenase, oxidase, hydroxylase. This name, these 4 name containing enzymes are here. Now we were talking about transferase. Okay, we completed oxidase, this oxidoreductase. We were talking about transferase. So transferase means something which transfers. The very easy, transferase. So transferase does the transfer function. Oxidoreductase does the transfer of electron that we have seen here. Here we classified the enzyme based on that. You can see these all fall under oxidoreductase. Oxidoreductase. Okay, and then transferase and other will fall under this group. So most of the transferase you can see fall under this group, and other group will also have this coenzyme. You can see the transferase. Let's see this. So transferase will transfer some molecule. That is why it is called transferase. So you can say transferase transfer a group from donor to acceptor, and that is how systematic name is given. First is donor name is written, then acceptor, then group. What is the name of this group? Group name is written, then you add after that transferase. So actually this is a conversion of this enzyme commission number. This actually enzyme commission number is also guided by this systematic name. You will see that. So you can see one name, enzyme name, very important, very, very important enzyme. The enzyme is ALT, alanine transferase, alanine aminotransferase. Okay, so this is an enzyme that transfer amino group. This is a common name, ALT. Okay, this is common name. Alanine aminotransferase is common name. So it transfer amino. This you can see here. You can see amino group. Here you can see this group is devoid of amino. So this is pyruvate. There is no amino. Now if you can see the same place here, this amino group is being transferred here. So this now pyruvate is converted into alanine. It carries amino group. And this amino group has been transferred from this glutamate to here, and now glutamate becomes alpha ketoglutarate. So this enzyme transfer amino group. This is ALT. So this is the one of the example of what transferase. ALT is transferase. You can see the same type of, this is ALT. This is ALT. This is ALT. So you can see enzyme commission number starts with 2. Okay, 2. And this is number given to donor. Like what type of group you transfer, actually it is related to that. So forget about this, just understand this 2 only. Okay, you don't have to, it is out of your scope to remember this. Just understand it is the batch number. You can see the number, enzyme commission number is number given to a particular function. So you can see 2 number will be with all this enzyme name. So alanine transferase is one of the example. The same type of other is aspartate transferase. You can see it also has 2 number and followed by other numbers. Very similar, aspartate transferase. Now here I have shown the example of aspartate transferase also. You can see this is aspartic amino acid, aspartate. It carries amino group and it is alpha ketoglutarate. So it does not carry amino. No amino group is transferred here. You can see amino group has been transferred. And then there is same place, you can see it is devoid of amino. So this is aminotransferase. We will talk in great detail about this aminotransferases in MD2. So these 2 transferases we talked, and this creatine kinase. You can see note this terminology, kinase. All kinases, the very important terminology, all kinases are transferase. All kinases are transferase. Okay? Will you remember this? All kinases are? Yes.
Transferase.
Remember, all kinases are transferase. All kinases are transferase. So we will talk about what is the function of kinase. All kinases are transferase. And then there is something called phosphorylase. Glycogen phosphorylase, very important enzyme. This is also transferase. All kinases are transferase and glycogen. These two are very important part of this transferase. These are very important. So let's see. This is pyruvate kinase. You can see. Again, I have explained. You can see this is refers to transferase. This is group. It transfers. This relates to group. It transfers, and then it relates to. You can see phosphotransferase with alcohol group. Acceptor. It talks about acceptor. You can see donor and acceptor concept. It talks about acceptor. It talks about donor group. It talks about acceptor, and then it is a specific name. You can understand. So all kinases are transferase. You can see what is the function of kinase. You can see this guy is kinase. So what is he doing? He is actually taking this phosphate group from ATP, and he's transferring phosphate group from ATP to some substrate. So that is why this is kinase. Did you get that?
Yes.
So kinase always transfer phosphate group from ATP to some substrate. Understand this. This is the kinase. So wherever you see kinase, whether it is protein kinase, any kinase, so kinase always does the same job. This is a very, very important. I am teaching you the name of only one enzyme, kinases, but you will see this kinase is repeating many times. Same concept will be repeated many times. You will understand the same concept. Okay. Whenever you see kinase, means this is a particular enzyme. Is what this enzyme doing is it taking the phosphate group from this ATP and transferring this phosphate group to some substrate. You have to understand that time. Whenever I say kinase, whenever somewhere you find kinase is written, so you have to understand that. Got that? So kinase, you can see the kinase can be, it can transfer this substrate, can be glucose, it can be protein. So based on that we can call it protein. If this kinase transfer phosphate from this ATP to protein, we call it protein kinase. Other group of kinase I have placed into substrate kinases, though the hair protein is substrate. Anyway, other substrate kinases you can see. So when it transfer, this guy transfer ATP, this phosphate from ATP to hexose sugar, glucose fructose, it is called hexokinase. When it transfer phosphate group to glucose, it is called glucokinase. When it transfer this phosphate group to phosphofructose, it is called phosphofructokinase. When it transfer this phosphate group to pyruvate, it is called pyruvate kinase. Just understand this kinase. Kinase is a very big group of enzymes. Even there in our body, more than 500 protein kinases are there. Protein kinases are there. And you may see there are kinases, if you count the number, maybe there will be 1,000 types of kinases. You will find more than 1,000 kinases in your body. So in one go, you are understanding all about those 1,000 kinases, properties of those this kinases, and these kinases are involved in various activity of your in your body. It is involved in metabolic pathway. It is involved in regulation of cellular mechanism. So this is very, very important kinase. You can see myosin light chain kinase. It is involved in smooth muscle contraction. So kinase is very important. That is why we have to focus on kinase. Now, second enzyme, this is very enzyme. When are you in fasting condition? How many of you are in fasting condition? Okay, if you are not in fasting condition, most of them, sometimes you will be in fasting condition. In fasting condition, this enzyme is very active. Phosphorylase enzyme. You have glycogen in your liver, and this enzyme breaks down glycogen. And by transferring phosphate group, you can see this man. Okay, this guy is actually cutting this bond by using phosphate group. So you can see it like X. So it is breaking this bond by phosphate group. That is why it is called phosphorylase. And then phosphorylase falls under group of transferase. Remember that. And we will talk about this phosphorylase in MD2. Here you can see glycogen phosphorylase breaks this bond between two glucose by adding phosphate group. This is glycogen phosphorylase. So we have done with transferase. Let's talk about hydrolase. And very good thing about this hydrolase, you already know many hydrolases. So you don't have to remember many hydrolases, because all digestive enzymes breaks the bond utilizing H2O, and that is why they are hydrolase. You can see all digestive enzymes. So you know the name of digestive enzymes. These are the digestive enzymes. These are hydrolase. So there can be non-digestive enzymes as well, which are hydrolase. So the important one are acetylcholinesterase. You will study about this acetylcholinesterase, very important enzyme. You will study about this in physiology. And then if this enzyme is inhibited, organophosphorus can inhibit this enzyme, and other drugs are there that can inhibit this enzyme. This enzyme is very important. That is why I have shown this enzyme. This is a hydrolase. This is a hydrolase. You can see this hydrolase. This is a hydrolase. Acetylcholine will be broken down into acetic acid and choline. So this is what this enzyme does. Now alkaline phosphatase, ALP. You can see ALP breaks, as the name suggests, phosphatase. It removes phosphate from organic phosphate. You can see phosphate is present in organic compound. It attacks here, it cleaves this. So organic and then phosphate. So it becomes phosphate and organic alcohol. So that is why that is the function of this phosphatase, alkaline phosphatase. And this is very important enzyme. This is very important enzyme for bone health. And also, this is also a tumor marker. And very important enzyme of liver function test. So whatever I have written, so this written, whatever you'll find written here, so go through all, memorize the enzyme names. Okay, go 2, 3, 4, 5, 6 times. That will not waste your time. If you go through this enzymes chapter, it will not waste your time. It will be very, like, every time will be like paid. If you go through the biochemistry topic, every single second will be paid. You will feel it will be paid. So go through each and every enzymes, memorize each and every enzymes for the time being, and then you will see it paid when you study biochemistry later. Lyases. What is lyases? Lyases means simply breaks the bond. Breaks the bond. You know, hydrolase was also breaking the bond. Hydrolase is also breaking the bond, but hydrolase utilize water. Okay. Then you can see transferase. This is also breaking the bond using phosphate by transferring phosphate. But this becomes transferase. This becomes hydrolase. But lyases has a different thing. Lyases does not utilize anything. It breaks the bond like that. Okay. Lyases does not utilize anything. Utilize anything to break bond. To break bond. So you can see this lyase break, and it always breaks a single bond. It does not break double bond, single bond. Single bond. Okay, so it will cleave carbon-carbon double bond. It can cleave carbon-hydrogen, carbon-nitrogen bond, and it will cleave this bond without utilizing anything. That is why this is called lyase. You can see aldolase. What is it? These are the example of lyase. Sorry, I told you not single bond. I told you there is one confusion. Okay. So whatever I told you, it does not utilize. Remember, it does not utilize anything to break bond. That's good. It does not utilize anything to break bond. And I told you single bond. No, it can break. It can be involved in double bond also. So that is the one thing that I deviated from. Double bond. Okay. When double bond is broken, it is also lyase that will break double bond. Understand that. So it will break bonds between carbon, 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 oxygen, carbon, nitrogen. Okay, these are lyases. We have two examples of lyases. You can see aldolase and you can see aconitase. Aldolase in glycolysis. So you can see here aldolase breaking the bond. This is glucose molecule. So you can see pink part and this black part. This will be broken down into two. So this molecule will not utilize anything, just break this molecule into two. One is glyceraldehyde, other is dihydroxyacetone phosphate. Glyceraldehyde 3-phosphate and other is dihydroxyacetone phosphate. Now here you can see citrate in Krebs cycle. You can see this aconitase enzyme, it is utilizing water here. So you might be confused that this is a hydrolase, but this is not a hydrolase. When water is utilized to break the double bond, it is lyase. This is the confusion between this. Hydrolase, here I will have to tell you that here hydrolase always breaks single bond. Okay, that I had to tell here. Hydrolase always breaks single bond, sigma bond. You can call it sigma bond between carbon, oxygen, carbon, nitrogen, utilizing water. You understood about hydrolase? Always breaks single bond utilizing water. But if double bond, this pi bond is hydrolyzed, double bond or pi bond is hydrolyzed by utilizing water, it is called lyase. Only this place, this will utilize water, and then it is called lyase. And which lyase? Hydrolyase. Did you get that difference between hydrolyze and lyase?
Yes.
You are confused?
Nope, not confused for me.
Okay, okay. If there is any confusion, let me know for other people. So this is lyase. There's hydrolyase will break pi bond, and hydrolase will always breaks sigma bond. That is the sigma is single bond. Pi is double bond. Double bond is called pi bond. Understand this. So two examples for lyase. One enzyme exists in glycolysis, other in Krebs cycle. Okay. Now isomerases. There are many types of isomerases. You can see the isomerases are 1, 2, 3, 4, 5 different types of isomerases with different job. So you can sometimes isomerase is given number 5, and the second number whatever you give number, it specifies this type. Okay. So racemase, epimerases, cis-trans isomerases, tautomerases, mutases, and cycloisomerases. These are the types of isomerases. You write is when this second number designate this type. You don't have to actually memorize that. Just understand that thing. That's all you need to know. Now you can say, let's talk about this isomerase. What is isomerase? We will talk about this isomerase in glycolysis. What is phosphohexoisomerase? Phosphohexoisomerase, second enzyme of glycolysis. Second enzyme of glycolysis. You can see this phosphohexoisomerase. Here you can see glucose 6-phosphate is being converted into fructose 6-phosphate by this enzyme. That is why this is isomerase. Isomerase carries first number 5, and then second number is 3 based on this isomerase, because it is a functional isomerism. It is converting functional. So this 3 number is given to this properties. One of this. It represents one of this. You don't have to actually specifically know which one is. So this is phosphohexoisomerase is an example of isomerase. Okay. You will see wherever you see this name, the enzyme come with this name, isomerase. The enzyme come with name with mutase. We will talk about, there are mutases in glycolysis also. So wherever you see the name come with mutase, wherever you see the name come with isomerase, generally we will see these two only in our biochemistry. So these are all isomerases, fall into group 5. Okay. Now ligases. Ligases, you can see this this guy joins the bond. Lyase breaks the bond. This joins the bond. And how does this join the bond? It always requires energy to join the bond. You can remember like joining this two rod by, you must have seen the joining of soldering of this two rod by using energy. In garage, somewhere you must have seen the joining of these two rods, electrical rods, soldering of rods by using energy. You can understand that way ligase. So ligase does soldering of two elements. So when you remember that, you will automatically you will understand that joining of bond will require energy. So ligase will always require ATP. Understand that. Ligase will always require ATP. Like soldering requires electrical energy and some other metals. So here this ligase will always require ATP. That is a very important concept. No ligase cannot join bond without ATP. So let's see a few examples. So coupled with hydrolysis of diphosphate, the ATP, bond of ATP, you can understand that. Now let's see this. This is systematic name XY ligases. You can see, we will see the example. A very, very important two examples are very important. That is why this, I told you, this classification of enzyme and example of this classification are very important. More you revise, more benefit will be for you. So you can see this enzyme, there are 20 enzymes, 20 specific enzymes for 20 amino acids. You will study this in genetics. So this enzyme is ligase. What does this enzyme do? This enzyme actually, what it does is it takes this amino acid and add this amino acid on a particular RNA. Let's see this enzyme, amino acid tRNA here, tyrosyl tRNA synthase. So you can see this tyrosyl tRNA synthase. What is tyrosine? You can see tyrosine tRNA synthesis. What it does is it adds this tRNA to this tyrosine to tRNA. This is tRNA. You can see this is tRNA will look like this. And there will be a CCA side chain here. Amino acid will be joined. So this enzyme will add this amino acid to this tRNA. That is why this is called ligase, by utilizing ATP. You can see clearly. You can see ATP, AMP. So 2-phosphate bond is utilized to add this amino acid to tRNA. So this is a very important, the beginning of protein synthesis. This reaction is start of protein synthesis. Remember, this is a very important example. Second example, I will be talking about this because I'm not teaching you, I will not be teaching you genetics, but you will see this reaction in genetics. And Dr. Guri will teach you in translation, protein synthesis. But I will be talking about this carboxylases, many of them. That is why I will ask you a question from this. This is very important. I told you one thing, if you can remember this. I told you today also. I told you to remember biotin. What does biotin do? It transfers carbon dioxide. So you can see it adds carbon dioxide to molecule. Biotin is part of ligase enzyme. I'll show you now. So you can see here, this enzyme is pyruvate carboxylase, because this carbon dioxide, what does carboxylase means? This enzyme is adding CO2. That is why this is called carboxylase. And this enzyme is adding carbon dioxide to which molecule? Pyruvate. That is why this is called pyruvate carboxylase. Now when it adds CO2 somewhere here, so this is pyruvate part. So it will add CO2 somewhere here. And if it adds CO2, it becomes other molecule, oxaloacetate. So because it is adding this carbon dioxide, I told you the soldering phenomenon. So you can see the ATP is being utilized here. And now here It is not shown, but remember, biotin is there. Wherever carbon dioxide comes, biotin will follow. Wherever you see addition of carbon dioxide to molecule, biotin is follow. This is, remember, this has universal rule, almost universal rule. So when C comes, B will follow. When CO2 is added, biotin will come. Okay, and this all will happen through this utilization of ATP. So A, B, C are coming together for this reaction. ATP is being utilized for soldering of the CO to this pyruvate, and biotin is also coming to help this soldering. Did you get this concept of pyruvate carboxylase?
Yes.
Will you remember that all ligases requires ATP?
Yes.
Yes. Just remember the soldering phenomenon. Which if somebody solders to iron rod, you put energy. So that is what this ligases is doing. You will study about DNA ligase. DNA ligase in genetics. There also you will see the two DNA molecule will be joined here by ligase by utilizing ATP. So you have to automatically understand that if something is ligase, it must utilize ATP. Even somebody does not tell you anything, you must understand yourself. It must utilize ATP. And sometimes what happens, you confuse between this ligase and sometimes you confuse between this kinase. Many students come with this. Oh, kinase, all kinases are utilizing ATP. ATP you can see. But here you see kinases is utilizing ATP. How? It is transferring phosphate group here. Mainly, rather than energy, it is using this phosphate group to transfer. That is why this is utilizing ATP. Okay, but here ATP energy of direct energy of ATP is utilized by ligase. So you can energy of ATP is utilized by ligase for soldering, for forming bond. Here difference between, what is the difference between lyase and ligase? The very important difference, ligase always utilize ATP, lyase never utilize ATP. You can see always utilize ATP, it never utilize it. You got that? The very important difference between, sometimes you can have confusion between ligase and lyase. How? I will tell you how. So you can see this is reversible reaction. The same reaction, if you see in this direction, it is breaking reaction. And the same reaction opposite, if you see, it seems to be a ligase type of joining of this two bond. But you don't find anywhere ATP. No ATP. And that is why we call it lyase. Understand this. Understand this very important difference. So I want you to go through all this classification twice, thrice, 4 times, whatever times you want. Then it will be very helpful for you. Memorize those all things. Then it will be paid. When you study biochemistry, you will understand biochemistry, and it will be paid in terms of understanding. Okay. So enzyme terminology, this is the part of snapshot of your first aid. So you can see kinases has been marked. That is why I've kind of focused on kinases. Here you can see phosphorylase has been given. That is why I have written phosphorylase. You can see this. Carboxylase, you can find carboxylase also here. Okay. You can see hydroxylase. So now you can understand the terminology. Dehydrogenase you can understand. You can understand this mutase. You can understand this phosphatase being present in alkaline phosphatase, ALP, phosphatase. So I have discussed all of this. Okay. Synthetase and synthase I did not tell you about, but you can read from here. Generally, sometimes synthase can be transferase, or this can be transferase or ligase. Okay, synthase can be transferase or ligase, one of these two. So let's do this question quickly. Which of the following group of enzyme always require coenzyme for their action? Yes, try to be quick.
A.
Very good. Very, very good. A is the answer. Always require coenzyme. Always require coenzyme. Okay, there is no any, like there is no any exception, always require coenzyme. Very good. Now let's talk about the second question. Which of the following group of enzyme is involved in production of ATP for survival?
B.
Yes, you have to grab concept from our introductory chapter. I told you, from plant, from sun energy flows to plant, from plant energy flows to us human. If you are taking bread, you are taking bread, then it comes here. If you are taking meat, you will be secondary consumer. Then what is happening? This photon is coming, and this photon is here in the form of glucose, and this glucose will be oxidized to release energy. Okay, so energy production, actually oxidation reaction is very important in energy production. You know, the whole mitochondrial inner membrane electron transport chain is nothing but oxidation reduction. So understand this, that is the concept. And other enzymes are also playing role, but the oxidation is very important for production of ATP. The name itself suggests we have know the oxidative phosphorylation. Oxidative phosphorylation. You can see phosphorylation happens because of oxidation. So this one terminology explains this question, oxidative phosphorylation. So phosphorylation happens because of oxidation. Which of the following group of enzymes are involved in digestion of food in the intestine?
C.
Yes, C, hydrolase. Very good. Now kinases fall in which of the following group?
B.
B. A? Is it A?
B. Transferase.
Yeah, it's B. You can see, I'll show you. If you have confusion, I will show you. All kinases are transferase. Okay? Transferase. All kinases. You can remember this cartoon for kinase. Cartoon, why I'm making cartoon? I'm not very good at computer so that I can make cartoon on computer, but I've drawn this cartoon. Why? In order to show that this person transfer phosphate from ATP. Understand this ATP. I have also animated this ATP so that you will remember the structure of ATP subconsciously, rough structure of ATP. I don't want to be specific about structure, but if you remember structure, you will remember concept. Okay, so you done hydrolase, kinase, all kinases are transferase. Now what is the role of enzyme named with kinase? We have talked about this. Transfer what? What does kinase do? Anybody, please be quick.
Transfer glycogen.
Does it transfer glycogen?
Transfers phosphate group.
Yeah, again, I can focus on this picture, this cartoon. What is this guy doing? This is taking this phosphate from the ATP and putting onto the substrate. So transferring phosphate from ATP to substrate. Understand that. Then what is the role of enzyme named with phosphorylase? You can again tell me. I have again second cartoon for this. So wherever I've made cartoon, those are important part. You can see this man, this guy is taking the X. Okay, X is very similar to phosphate, phosphorus, PO4. Okay, PO4 group. So phosphate, and then breaking this bond between this glucose. So transferring this phosphate group to this and breaking the bond. This is the phosphorylase. Understand this. This is the job of phosphorylase. Just look at this. Okay, understand this too. Every questions I've given are very, very important question. Most of the carboxylases in body are part of which of the following group? Yes, be quick.
D.
Ligase.
It is D, ligase. Carboxylases are ligase. Carboxylases are ligase. I will show you the carboxylase. I told you ligase. Ligase are the enzyme that is shouldering the two bond. So you can see here there is pyruvate, and it is CO2 has been added here, and it requires energy. ATP is being utilized, and biotin. All ligases utilize biotin. Right somewhere. It is very important. Biotin. These are all carboxylases are biotin containing enzyme. Write somewhere else. I have not written. I will. So you write yourself. Biotin. Write somewhere. Okay. Did you all write somewhere? Carbox require biotin. Yes. I'm going to ask this question. 100%. One question is for sure. Take this. Note this. I'm going to ask this. Definitive one question. Let's see. I have given you one question. Okay. One question is out.
Sorry, what's the question?
This is the question. All carboxylases require biotin.
Oh, okay.
All carboxylases require biotin, and biotin transfers CO2. Add CO2. CO2, biotin, and carboxylase. Carboxylase itself means carbon dioxide addition. So I am going to ask this question definitively. So you already got 1 marks. If you focus attention, you already got 1 marks. This is because why I'm telling you this question. I'm telling you question in the beginning because this is very important. I want you to memorize anyhow. You got that? This is very important. This reaction, the name of enzyme itself is very important. Pyruvate carboxylase. This reaction is very important. And that is why I'm told you this question I'm going to ask definitively. Definitely, I'm going to ask this question. So, which of the following group of enzyme always utilize ATP?
Ligase.
Very good. Ligase. Utilize vitamin as coenzyme. Carboxylase utilize which of the following vitamin as coenzyme?
Biotin.
A definite question. This one of this, like from this concept, one question will come from pyruvate carboxylase. A definite one question. I can ask my own way, but a definite question from this. Okay, so carboxylase, we have done this. Now let's talk about factors affecting enzymes. Okay, so this is a very easy thing. I told you in the beginning only reaction condition. Here, this guy, this very efficient, very productive guy called enzyme require a particular temperature and pH. So this is factors affecting enzyme. So if you increase temperature, you will affect the activity of this. If you change the pH, you will affect the activity of this. So these two parameters are very important. So these are two factors affecting enzymes. You know, these are factors affecting enzyme. You can see this. So enzyme concentration, these three are there. Okay, these three are there. So let's not be concerned more about this. Let's first talk about this and this: temperature and pH. So there are 5 factors. I have to write those 5 factors. But more important is temperature and pressure. Substrate concentration, enzyme concentration, coenzyme concentration. It is related to enzyme kinetics. Enzyme kinetics. It is not that it is not important, but we will talk in them in enzyme kinetics. It is part of enzyme kinetics. Okay, these 2 are most important factor here. We will ask question under this topic from temperature and pressure. You will see. Let's first talk about temperature. And it is also very easy. You can see both have this bell-shaped curve. So you don't have to memorize too much. So both have temperature and pH, both have bell shape. What does this mean? There is a fixed temperature point, and there is a particular temperature point, and there is particular pH where this enzyme is most active. This is velocity, activity of reaction. So this is the place where the enzyme is most active. Optimum pH. This is called optimum pH and optimum temperature. This is the place. This is the temperature and this is the pH where enzyme is most active. You understood? Most active. And you know, when you decrease the temperature from this, then also activity will decrease. When you increase the temperature, then also activity will decrease. And that is what this is, a bell-shaped curve. Y-axis, you can remember Y. This is Y. So you remove this, this becomes V. So you never confuse what is there on Y-axis. Y-axis always has velocity. If you add this, it will become Y. So Y-axis always have velocity. So if you, in both conditions, if you decrease or increase temperature, the activity will decrease. This is what temperature means. Now there is one very important question here that will come. So this is also a question. What type of curve do you see in temperature? If you, what type of curve do you see in relation to temperature and activity of enzyme? So this is bell-shaped curve. Temperature coefficient is second very important question. So you can see, you can read this. What is this temperature coefficient? The number of times the velocity increases. The number of times this velocity increases when you increase the temperature or change the temperature by 10 degrees Celsius. If you change the temperature by 10 degrees Celsius, you can put the curve here. So when you change the temperature, like here is, you can see here is some, and if you change, it's like this. Here is one velocity, V1. Here is second velocity, V2. So if you change the temperature, you see how many times this velocity has changed. Generally, the velocity changes by 2 times. V1, when you change the temperature 10 degrees Celsius, the velocity changes by 2 times generally. And this is called rule of Q10. And that is why you put, if you put food in warm temperature, food is spoiled faster because bacterial activity is higher at higher temperature. So this is the rule of Q10. When you increase the temperature by 10 degrees Celsius, your velocity of reaction will increase by 2 times generally. You got that? Did you understand this?
Yes, sir.
Now, why does this happen? Why if you increase temperature, too much temperature, or decrease too much temperature, the velocity, like activity, will become 0. Why does that happen? Because when you increase the temperature, or if you decrease the temperature, there will be denaturation of protein, because enzyme is a protein. So enzyme will fall sick. Molecular. The way you fall sick, the same way enzyme falls sick. And sick person cannot work properly. Sick enzyme cannot work properly. You have to understand that. Same goes for pH. Same goes for pH. So pH, if you decrease the pH, increase pH, anyhow, the enzyme here also will become sick. And you know, enzyme binds substrate. Substrate is also biomolecule. So a charge of, you can see, charge on enzyme, and sometimes charge, because charge glucose have also some charge property. So charge of substrate can also be altered. And that way, this enzyme substrate can fall sick because of change in pH, and because of that activity will decrease. You know, the enzyme activity is influenced by charge on the enzyme, because enzyme is a protein. There will be certain charge pattern on the protein. If you change the pH, pH is proton. If you change the concentration of proton, the charge on the enzyme will change. And if charge on the enzyme changes, the enzyme will fall sick, and that means fall sick means enzyme will be denatured. And then that is how the activity will fall either side of this optimal pH. Did you get that?
Yeah.
Temperature, pH is very, very important. Now I will take just 1 minute to talk about just enzyme concentration. If you increase the enzyme concentration, the activity will increase. So this is a straight line. If you increase the substrate concentration, so up to certain point it will increase. After enzyme, all the enzyme is occupied by substrate, then there will be stagnant. The activity of that reaction will be stagnant. Now you can see the coenzyme for prosthetic group. Prosthetic group, this will be the pattern when coenzyme is tightly bound. So it behaves like enzyme. So a straight line. If we can increase the concentration of prosthetic, it will like this. Here, when coenzyme is loosely bound, so it will follow the curve of this substrate. So this is this three factor affecting enzymes. So these two are more important. You have to remember, and these are very easy to remember also. They are very easy to remember. Okay, these are factors affecting enzymes. So is there any question? Yes, we have some questions also after that. So coenzyme which requirement increases with increase in protein intake, plays key role in glycogenolysis, loss of memory and confusion, are related to deficiency of vitamin B6. So you will study about this. This is related to vitamin B6. So vitamin B6 is very important for protein metabolism. Remember this and memorize this. Okay, this is, I have not taught you, just memorize this part of question. So person having delayed wound healing might have deficiency of which of the following cofactor? Zinc. Remember, okay, zinc, because it is part of, you'll see, it is very helpful in DNA, RNA and protein synthesis. Okay, and that is why if zinc is not there, so this all process will be compromised and wound healing will not be proper. So these two questions you can remember, just remember these two. And then a person cannot tolerate pH drop because below 6.8 because of what happens?
Enzyme denaturation.
Yeah, enzyme denaturation, and the enzyme becomes inactive because of like the charge activity over the enzyme is altered, and that makes enzyme denature. So this is the end of this class. We have little time, so if we can utilize little time, so I want to utilize 2 minutes, just 2 minutes, so that I will give you an introduction to next topic. This is the topic for today, but we talked about that. So anyway, you can see the enzyme kinetics. I will just introduce this topic so that we will have very easy. Enzyme kinetics and regulation are very, very important topic. So we'll talk about enzyme kinetics. Enzyme is very important topic. That is why I'm going a little slow in the beginning, because you are like fresh, you have joined the course fresh. So if you don't understand, you will have problems. So that is why I'm going slow. You can see I have summarized everything here. If you remember this chart, you will summarize everything about the enzyme. So you can remember it. This is enzyme. So S3, R3, you can remember this graphs, especially these graphs and especially these graphs to remember factors affecting enzyme. Now you can remember this like this topic, this what is this slide I have already given. You can, especially I have told you biotin here. You can remember this. So these are about teeth. So you can remember about teeth, active site, or reaction site. And then you can talk about regulation. We'll talk about regulation. You can remember regulation also, 3 types of regulation I've summarized here. So specificity. So you can see carboxylase, I have written here, ABC enzyme, you can see carboxylases, add CO2 here. Now phosphorylase, kinases, I have added here this 3 very important enzymes. And then we'll talk about this kinetics in this next topic. Then we will talk about comparison. So enzyme inhibition. We'll talk about enzyme. These graphs are very important. So you can capture this graph. Okay, just see this graph and imagine in your imagination. And you know, velocity always will be on y. Wherever you see, velocity will be on y-axis. And other thing will be on x-axis, but velocity always will be on y-axis. You can see anywhere. So this graph is summary. That is why I put in the beginning. So what does kinetics means? Very simple. Kinetics means speed. Just remember, kinetics means speed. So we'll talk about this part of enzyme, velocity. What affects this velocity of enzyme? Kinetics means speed of enzyme. Activity of enzyme, speed of enzyme. So why speed of activity of, like speed of enzyme means speed of reaction. It means speed of reaction. The enzyme responsible for speed of reaction. So how speed the reaction an enzyme can catalyze is kinetics. How speed the enzyme can catalyze a particular reaction, it is called enzyme kinetics. Okay, more faster if enzyme can, the kinetics is faster kinetics, slower kinetics, you can call that way. So why does this enzyme increases the rate of reaction? Because of enzyme lowers activation energy. So we will talk about this activation energy. I will give you the concept of activation energy, and I will move to this equation next class. This is very important equation. So let me do attendance.